What I'm going to say here is so important. Um, some people would call it controversial. It's not going to be controversial, it's going to be very scientific. But the implications of what I'm likely to say will not sit comfortably with those who have promoted a narrative. Now, people need to understand that I am not anti vax, but I have a lot of questions when it came to the pandemic and when it came to how we instituted our approach in the pandemic. And so I ask hard questions. And it's especially important because I've been doing this research now for over 4 years. So I'm going to take you through a few steps. And what I'm going to be showing you is this very, very important paper on SARS-CoV-2 specific plasma cells are not durably established in the bone marrow long-lived compartment after mRNA vaccination. It was published on the 27th of September 2024. This is in Nature Medicine, one of the top publications, so it's well accepted. That was a big paper for them to publish because the implications of that are humongous. Now, you are going to hear some things here that I will categorically say that I am sure no one has thought about yet. So remember this date, and we will see in time if my analysis is correct. And this is what I woke up with this morning thinking, goodness, I think this is the reason that this paper picked up such a very, very important piece of the puzzle. To get the context, you have to remember that we're now on to number 9 or 10 booster. And what they've always known is that the vaccine immunity was waning very, very quickly. What was the mechanism for this? Why was it that after each COVID vaccine, the antibody level would drop quite quickly within weeks sometimes, and by 6 months needs another booster, and the same thing happens again? And it truthfully didn't make sense from an immune perspective. But this paper here is now able to give us some understanding as to why it's occurring, or I should say, the mechanism that it's occurring. And then I will extrapolate as to why that mechanism is occurring, if that makes sense. So, right before I start, just a reminder here about Disease X. Are you prepared? Remember, when I talk about Disease X, I'm still talking about COVID. But in it, we are looking at ways that you can be prepared for what's coming in the next few months and years. With the threat of the pandemic, ongoing viral circulation, we got our Kickstarter program done, and now we want you to help us to get to number one on Amazon. That's going to be in a few weeks' time. But let's get back to this question about what in the world is going on with regards to the COVID vaccine. Now, many people have argued they shouldn't call it that. And actually, I'm beginning to reach the conclusion, based on some of this research, that some of those people may be right. Let me be careful here. So let's start with some basic stuff. As usual, I'm going to be showing you from my presentation slides a few thoughts that I have put together. And I will take you through just some basics here before I go back to the paper. So just remember the rules. This is the coronavirus. It's here, it's like a ball. And on the surface, you have sometimes about 25 of these. Blue here represent spike proteins. And this is the cut section of the virus. So the spike proteins are projecting out. This is what it uses to enter the cell. It's like the key to open the door to get inside the cell. Once inside the cell, it releases the nuclear capsid and the RNA, which then replicates over and over again. The natural immune system targets not just the spike protein, but the membrane proteins, envelope proteins, nucleocapsid proteins. And interestingly, it doesn't actually target the spike so heavily. It also looks for what we call ORF proteins, which are produced once the RNA strands start to be making proteins inside the cell. And this is essentially what a spike protein looks like. This is the side-on view. The receptor binding domain is here. Again, this is what it uses to bind and get inside the cell using ACE2 primarily. This part here is the S2 part. This is what sits in this lipid layer here. And you have about 25 of them on the surface of each virus. And that's the basics. I'm going to come back to the presentation in a second, but I just wanted to highlight some important points from the paper. This paper was done in the US. It was received on the 22nd of February 2024 and published on the 27th of September. I suspect they had to go back and forth with this one because the implications were so serious. So you see how long it takes sometimes for a paper to be published. And essentially, I've highlighted here because they were testing not just COVID, they were testing influenza and tetanus. And so they have highlighted here that they recognize that, as usual, you can't get a paper published without saying this. Vaccines are effective at protecting from severe disease. It certainly reduces the likelihood of it, but the protective antibodies wane rapidly, even though SARS-CoV-2 specific plasma cells can be found in the bone marrow. So they're trying to understand what is going on. And essentially, what they found is that while the serum in the bloodstream, IgG titers specific for influenza and tetanus, correlated with IgG for long term plasma cells, okay? Serum IgG levels for SARS CoV 2, which waned within 3 to 6 months after vaccination, were associated with none. Well, maybe a few long term plasma cells. Now, this is very, very important. So, just to give you a little bit of background here, when your immune system is faced with, say, a virus or a bacteria and it's trying to make a memory, it has plasma cells. So plasma cells are the antibody producing cells. They produce all the antibody, and oftentimes they could be at the mucosal linings, or they could be, say, in the lymph nodes or the spleen, and they're producing antibodies. But the body wants to remember the infection. And so a cohort of these plasma cells are then sent to the bone marrow, where they are then kept very safe and protected, and the body keeps them in case it has to face this infection again. And this is how you have the rapid response to the immune system after it has faced a virus. So, this is part of natural immunity. And so, what the researchers were doing is that they were trying to understand why was it that the antibody levels waned so quickly. And so they were interested in what happens with the SARS-CoV-2, the COVID spike protein mRNA vaccine, in terms of the bone marrow. That's really the question that they were asking. And important to note that when you look at this here, they highlighted here that tetanus vaccination, which we get every 10 years, generates antigen specific bone marrow, long term, long lived plasma cells, and affords safeguards for decades with a serological half life of 10 years. This is a normal vaccine. Okay. And one of the problems that we have now is that people are starting to think that COVID vaccines are the standard for normal vaccines. No, no, please don't. This is new technology. This is in a disease. I think they shouldn't have done it in this way. We cannot afford to let this cock up destroy the history of vaccines and the benefit of vaccines. Now, let's be clear, I'm also on the side that I think that possibly there is over vaccination, where there are too many vaccines being used in children, say. But that's a separate question. But the point is, the ones that are being used, the DPT, the measles, the mumps, the rubella, these are standard old vaccines, especially as some of the research had been done with the attenuated, the whole virus vaccines, tremendous benefit to the immune system. So I don't want us to get confused with this. And actually, this paper is indicating something that is so significant that think they need to go back to the drawing board. And so this is the bit that I wanted to highlight in the paper. I'm trying to get the image in the right place so that you'll understand what it is that they highlighted that was so concerning. So I've got an image here, and it was looking at the time frames. And you have to remember, it was a small study, so it wasn't a huge study, but they followed people, and some of them had up to 5 doses. So that would mean the two initial doses plus three booster doses. And they were looking and observing the characteristics as to what happened with regards to the bone marrow. And there is no other study like this. So I don't think anybody can debunk it easily. So here is essentially what they found. So this is looking at the timeline: 2.5 months, 14 months, 23 months. Then they're looking here at the non long lived plasma cells. So that would be like in the lymph nodes and the mucosal lining and so on. And then the long lived ones down here. So this is the long lived ones at the bottom. And then they were testing flu, tetanus, SARS-CoV-2, so to speak. Flu, tetanus, SARS-CoV-2. So all three. And you can see that at 2.5 months after, you can see straight away with the flu vaccine, you're having high amounts here in population B, and population D in the bone marrow is starting to have some as well. Similar pattern with tetanus. Actually, more are in the long-lived compartment than even in the non-long-lived plasma cell compartments. But here, you can see that therefore they're producing antibodies probably in the lymph nodes here, but zero in the long-term plasma cell. When they looked at 14 months, again, similarly with flu and tetanus, you can see these little blue dots here represent the cells. Nothing again by 14 months. 23 months, and this is clearly in the case where they had the booster, because you can see lots of antibody presenting, antibody secreting cells, but only one here. But look at the difference with the flu and influenza. So there is clearly a problem with regards to the fact that the immune system is not keeping these antibody producing cells in its long lived compartment. So, one part of the question is that it's happening, and that then explains why the immunity wanes so quickly, because the long lived plasma cells are not there. But then that made me start to think, and this is a bit you will not have heard before. Why would the immune system respond like that with regards to these mRNA vaccines? So, my rule of thumb is I always look for an analogy, because nothing in medicine is new. It always has happened before. And so it was bugging me. I was thinking to myself, there is going to be a reason why this is occurring. What could be the reason? Where else does this happen? Good. That's where I'm going to start you from here. Where else does the body produce an immune system response and doesn't keep long-lived immune cells? That was a question I had, and I think I found the answer. So here is what it is. Here we go. This is a gram-negative bacteria, usually like E. coli. These are the ones that cause UTIs and cause sepsis. They can cause really severe sepsis. And this picture is showing you a cell wall here. So it's cutting through this cell wall. And inside the cell wall here on the lining, They have a number of proteins there, but there is a lipopolysaccharide here. And when you break down the lipopolysaccharide here, you can see what it looks like. There's a lipid A, then the core polysaccharide, so lots of molecules here, and then repeating polysaccharides. And this is the lipopolysaccharide, which is present in the surface of gram-negative bacteria. Okay. Now, what happens is that these bacteria can then produce what we call exotoxins. That means that they produce toxins that will damage the cells. So the bacteria is alive and producing toxins, like Shigella or something like that, or cholera. Or when the bacteria is being destroyed, it releases endotoxins. Okay. Now, I go back again to that image. So the endotoxin that is relevant is this lipopolysaccharide, right? What then happens is when these endotoxins are released, so if the bacteria gets in the blood and it's being destroyed, so you have to imagine that the person has a urinary tract infection. The bacteria have gotten into the bloodstream, right? Um, Even if you're giving them antibiotics, sometimes they haven't had antibiotics yet, and then they get septic. They get really, really sick. And that's because the bacteria going into the bloodstream is picked up by the immune system. The immune system targets the bacteria, destroys them, but in the process of destroying them, it releases these lipopolysaccharides. These are endotoxins. So the bacteria is dying and so it's releasing these lipopolysaccharides. This is what causes the immune system sometimes to overreact because the immune system has some very, very strong responses. This is what it would look like. It has very specific binding proteins so that when this lipopolysaccharide is released and it binds here, it kicks off a whole series of immune responses with interferon, pro-inflammatory cytokines. And this is where the person can get extremely septic. And if you don't have enough time, they can die. And so this is what we call gram-negative sepsis. It's one of the most serious forms of infection that can occur. And the problem is the immune system overreacting to the endotoxins. And so they sometimes need not just antibiotics, but they need support on intensive care to get them through that period of endotoxin. Now, you would think that because of the significance of this endotoxin being released, that the immune system would want to remember it. No. Literally, every time the immune system faces this endotoxin, it does it as if it has faced it for the first time. Now, think about that. It doesn't want to have the memory of this endotoxin. Different sometimes for an exotoxin, but for the endotoxin, it doesn't want to remember it. And so I had to think, why would that be? What really is the mechanism that would cause that to happen? The problem is that these endotoxins can trigger lots of problems for the body and for the immune system. And this is what it can look like in terms of what can happen if these endotoxins continue to be triggered. You can end up with chronic inflammation, leading to heart disease, neurodegenerative diseases, autoimmunity. These long term antibodies may then target self tissues. You can have immune tolerance and suppression, immune complex formation, because then the antibodies may bind to toxins and then get stuck in the kidneys or in the bloodstream, leaky gut syndrome, increased risk of sepsis. It damages the gut microbiome imbalance, and it can cause problems in terms of endotoxemia, ongoing toxin production, and immune response, leading to all of these things. So the body, even though it faces this really serious infection, it doesn't keep long term memory of it. And this is therefore what happened here. When you look at it again, up to even 23 months, it's almost fighting. Don't want to remember this. Don't want to remember this. Switching it off. This is the long term compartment in terms of the spike protein. Doesn't want to remember it after 2.5 months. Doesn't want to remember it after 14 months. Being forced to remember it after 23 months, but still trying to prevent the immune system from recalling this specific trigger. In my mind, what that means, and this is why it's so important to reflect on, in my mind, that indicates that the immune system is treating the spike protein as a toxin. It's not treating it as a protein, as something that it can come across. It is treating it as a toxin. It doesn't want to remember it because of the risk of autoimmunity. This is what I said from the start. Severe COVID 19 is a viral mediated autoimmune disease because the spike protein is like Velcro. It sticks to everything, it sticks to so many proteins. And so the immune system is saying, I cannot afford to remember this protein because it will make the body sick. Well, just think about that. Just reflect on that and think, what in the world is the scientific community thinking? Really? I mean, this requires going back to basics. My suspicion is this has to do with the spike protein, not necessarily mRNA. But they need to know if the same thing happens with mRNA. Suppose we find that there is no long lived antibody response after mRNA because of some kind of strange mechanism with the immune system. This is extremely important. And I would say this is so important that I don't know how public health will be able to justify continuing with their current strategy, not answering this question. It, it just means effectively that no matter how many, you then have to literally accept that every 6 months or every 4 months, I don't know how long it takes for the immunity to wane, you just keep on going. Think about the guy who did 215 vaccinations. I bet you after 6 months, if he stopped, he still wouldn't have long lived plasma cells. That would be the case. That would be the case that they should look at. If this guy, after 215 vaccine doses in Germany, he took that, if he didn't have long lived plasma cells, you are beating a dead horse. It's never going to get up, it's never going to run. And what are you doing? So, this, I think, is probably the most significant finding, in my view, that we have come across is that long lived immunity in the bone marrow is unlikely to happen no matter how many doses are given. And so, therefore, you should come back to the question, if the immune system is not trying to remember it, why are you trying to push it? That's really the question. So I hope that this stays around because it's asking a very challenging question, but it is based on the research. I'm not making this up. And so I think it's an important point for people to reflect on from the scientific community. I don't think there's going to be a good answer that is going to be put out there. I think that this requires them going back to basics and trying to figure out, okay, what are we doing here? What is the reason for doing it if we are never going to get long lived plasma cells and we're never going to have long lived? This is not about the variants. This is about the fact that the immune system refuses to remember the spike protein. So, challenging times ahead. No easy way out of this because now what it means, effectively, because they have killed any other option, there is nothing left. It means, therefore, theoretically, there is nothing left. All the eggs were put in one basket and the basket has crashed. What do we do? So, as a reminder, yes, in the link below, please remember to join us with regards to disease X. There is a link as to strategies for the pandemic. I think this is going to become more and more important. You really have to start thinking outside the box because I think we're going to be facing challenging times ahead. So, I'll keep on keeping you updated as long as they will let me ask the hard questions. Have a great evening.